a talk coming up next month, right, Celine? So you and Cynthia are going to be doing. I am so interested in that talk because both of them have moved into private practice, and they're going to share their gems of wisdom that so all of us, when the academic systems fail and the world goes to crap, we are all going to open up private practices. And Celine and Cynthia are going to teach us how to do that next month. Right, Cynthia? All right, Cynthia, Celine? So that will be a really good one to do. And then let me look at my sheet here because then after that, in March, we have Steve Scheinkopf coming on. He's going to talk, he does lots of really cool research on early cry and some other early markers that he's been doing for many, many years out of Rochester and here in Missouri. So he's going to be talking. In April, we have slotted Jamie Winter. She's one of the world's recognized ESDM NDBI therapists, just to give kind of a talk on what that looks like in the state of the field with regard to that intervention. But I am totally excited to have Jamie here, and in full transparency, I invited Jamie several months ago and then forgot that I invited him. So I invited someone else, never got the end of it on text, was shamed completely, was reminded of this scene in Game of Thrones where shame, shame, that was me after I did that. So Jamie, I apologize publicly for that miscommunication on my part or error on my part. But yeah, Jamie, I've heard him talk before. He's a fantastic speaker. No matter what he talks about, he conveys it with passion, caring about the families. But above all else, Jamie's one of the premier scientists that we have in our field and is doing literally groundbreaking work that we should all be aware of. So I was really, really happy when he said yes to coming and talking to us today on IDEA. So Jamie, no further ado, on to you. I will turn off my camera and my sound. So
Thank you very much, Steve. Thanks everybody for joining. Nice to see many of you, to meet some of you. Okay. All right. And are you seeing my slides appropriately?
yep, that's the right one.
Okay, great. So Steve said lots of nice things about me. He didn't say what I do. And so I'm a child clinical psychologist, and I'm still kind of clinically active, although I shouldn't say it, maybe say that out loud. I shouldn't be own that in this group. I'm technically still clinically active. And then I'm also a cognitive neuroscientist. And really what I'm going to talk about today is really the work that I do as a neuroscientist in biomarker development and the aspirations that I have to make these biomarkers, these indices that we study in the brain, useful for the work that I and many others do clinically. These are the groups that support my research and me, and I don't think there's any conflict with the things that I'll talk about today. And this is kind of what I want to talk about. When I say biomarker, what do I mean by that? What are biomarkers? Both in terms of the way the FDA operationalizes them because they take it seriously, and then some of the more colloquial ways that I think about it, both scientifically and practically. Then I'll tell you kind of my own personal narrative in this domain, work that I've done over the course of the past 20 years looking at a particular biomarker, an electrophysiological brain index of face processing called the N170. And I'll kind of walk you through some different phases of this work from kind of coming to think about this as something that could be relevant to exploring it to understand more of its properties to then thinking about how, well, can we measure this with such rigor and certainty that we could qualify with the FDA so it could be a tool that anybody could use. And then I'll kind of wrap up by talking about some of the kind of things that are in the works, moving this work into areas that are more ecologically valid, presumably, translating it into direct treatments, and then expanding the population, and then talk about some different kinds of biomarkers that we're starting to try to develop using behavior. Okay, so really, to begin, this is the problem that my work tries to solve, is that when I am being a neuroscientist, I have many, many different tools at my disposal to help me understand how the brain works, how it works in autism, how the differences that I see might be useful in understanding how to help autistic people in an abstract, kind of basic, non-applied way. But when I try to do these kinds of things in an applied setting, I only have one tool, This literally, the technology hasn't advanced since Leo Kanner was evaluating people with autism. We rely on our eyes. Um, I'm sure many of you experience some of the same kind of quandaries. There are problems, I think, with this. I want to acknowledge it's actually remarkable to me how good we are at using the human eye to quantify behavior in autism, social behavior, which to me is particularly abstract, ephemeral, and hard to put a finger on. We've gotten so good at it. We have tools. We can be pretty confident that a clinician in New Haven is going to do a pretty similar job about rating something as abstract as, the nature of a social overture to a clinician across the country. But there are inherent things that we're going to run up against in when we rely on the clinical eye. You know, no matter how we scaffold or constrain our insights with standardized tools, our insights are inherently subjective. We see what we see. The nature of clinical assessment is that we have a finite period of time in which to observe human behaviors, and because humans behave in so many different contexts and we bring them to a strange place and make them do strange tasks, it's hard for us to know what we're quantifying, what we're seeing, and whether it's reflective of things in other contexts. And then there can be other factors too that we may not be aware of, but we could be introducing systemic biases. This is a chart from a paper recently written by Maggie Azu and Gloria Hahn. And what they did was they looked at this differences in what the eyes of clinicians saw and what the eyes of parents saw. And what they found was that relative to parents, clinicians systematically overreported for males and underreported for females. So it might not just be that we're missing things, but we could be seeing things that aren't there or aren't different. And so I think that a useful way, a useful supplement to our reliance on human observation would be to have more biological kinds of information to introduce. And so we would call those things biomarkers. And this is how the FDA defines a biomarker. When I say they take it seriously, what they've done historically now for maybe they started in 2015. No, before then, but they convene a group of scientists, the FDA and NIH, really to think about this, how we should define and operationalize biomarkers. This is their working definition, which I won't read out loud to you, but suffice to say, it's a pretty broad and generic description. Really, if you think about it, behind all that jargon, it's just saying these are things that we can measure that are reflective of biology, whether it be adaptive or maladaptive biology, static or dynamic biology, we're measuring things that are biological. That's the FDA's term. The FDA also has different categories of biomarkers. I think this is something that's very, very important for clinicians and researchers in autism to think about. Because I don't know if this is true for many of you, but often when I say biomarker to a person in the field of autism, what they hear is diagnostic tool. And it doesn't have to be a diagnostic tool. And to think of it in that way is really limiting in terms of the potential utility of biomarkers for clinical work in autism. So a diagnostic tool is one of the ways we could think about biomarkers. So in autism, this would be something that we would measure that tells us whether a person falls into this category of autism or not. The FDA talks about a category called susceptibility, which historically in autism would have been thought of as like at risk. More presently, we would say like increased likelihood, but telling us that someone is likely to end up in the spectrum in the future. There's a third category called pharmacodynamic or response, and this is telling us something about the response of what's being measured to a treatment. Is, for example, if we had a treatment that we thought was going to impact certain neural systems, do we see those systems activating or changing in response to the treatment? A prognostic biomarker is one that would tell us the future, how a person might look. Maybe if we were to measure a prognostic biomarker in a 3-year-old with autism, it could tell us something about what we would expect for their language development or their cognitive development down the road. A predictive biomarker is similar, but specifically in the context of a treatment. The idea that it, it's telling us something about how a particular person will respond to a particular treatment. And then there's another kind of biomarker that actually isn't named by the FDA. The FDA thinks of this as a kind of diagnostic biomarker, but a stratification biomarker. You could think of it as diagnostic of a subgroup. The idea that we have this really heterogeneous group of people with autism, but we might have something that we could measure that would break them down into more meaningful subgroups, which would be something that would be really useful for us. I think that this is also a really great example, too, of some of the shortcomings of clinical inference alone. We as a field did this. In DSM-4, we had subtypes of autism, and then they didn't work, and then we backed off of them. So we still remain in search of these kinds of subtypes, and our clinical eyes haven't delivered so far. We think when we think about a biomarker, we think in FDA terms about what's called the context of use. And this is basically one of these categories, but then a purpose. So, for example, it might be a stratification by biomarker for selection for enrollment in a clinical trial. So that would be the context of use for a biomarker. These now are the kinds of things that I've thought about outside of the FDA's work, really. But if I were to try to figure out whether a potential biomarker could be useful, what would I want to know? Irrespective of whether it's a diagnostic biomarker or not, in principle, I'd want to understand whether it hangs together with diagnosis. My computer is so hypersensitive to gesture that I don't even know. I'm not even going to try to figure out how to put down my own hand because it'll come back up. Just think of it, I'm waving to you all. I'm glad that you're here, and that's me saying hi. Um, if it's not hanging together with diagnostic status in some way, we might be barking up the wrong tree. Doesn't need to hang together with diagnostic status. Perhaps we want to see biomarkers that are associated with specific functions. We might think of a profile of biomarkers. One could be telling us something about auditory perception. One could be telling us something about sensitivity to touch. For those biomarkers, we wouldn't necessarily it wouldn't be so important how they hang together with diagnosis, but we would expect them then to vary with other measures of function in those domains and in ways that are specific. For example, if we had a biomarker that we thought measured speech and language, or say like measured speech production, but it was actually measuring IQ, it would be hard to tease those apart because IQ is going to put a ceiling on speech production for many children. But if we had a treatment that improves speech production, we might not see the biomarker, which is actually measuring IQ change. And so we want to know what things measure, what they don't measure in a dissociative way. In autism, when we talk about a person with autism, we don't know if we're talking about someone who's, 3 years or 30 years old. And we wouldn't expect the brain of a 3-year-old to work or look the same as the brain of a 30-year-old. And so we have to be thoughtful about whether the biomarkers that we're studying are specific to a particular developmental range or they can be used across the lifespan. We also need to be thoughtful about how the process of measuring a biomarker could affect its values. This doesn't matter for everything. Like for a genetics biomarker, when we're doing a blood draw, it's not really important whether the child likes the blood draw or is uncomfortable during the blood draw. The information is going to be the same. When we think about measuring brain function, a child in acute distress during an EEG recording is only going to give you information about that distress, not about the different things that you're trying to measure. So that's really important to understand. And then lastly, it would be nice to know for a given marker whether it tells us something about clinical status. As a child gets better or gets worse, does the biomarker track with that? I also think that there are practical considerations. Oh, I want to highlight that if we think about all these different kinds of potential attributes, a biomarker doesn't have to do all of these things. And it might be less useful if a biomarker did do all of these things. Like if you had a biomarker that you thought could be applicable in diagnostic contexts of use, you would absolutely need it to be sensitive diagnostic status. But whether it changes as a person gets better in treatment may or may not be useful and could potentially get in the way of its use. Another example, if we had a biomarker that we thought might be a response or pharmacodynamic biomarker, there, the most important thing would be that it's sensitive to change in clinical status. And so it's really important to acknowledge the nuance in biomarker development. The information that we gather to evaluate the utility of a biomarker is really context and purpose specific. And a biomarker that may not do some of these things well could do others of these things very well, and that might be enough for it to be something that could be very useful in clinical research or clinical practice. There are also practicalities relevant if we actually want to think about deploying biomarkers in meaningful ways at scale. It has to be something that we can do in clinical populations or in children. This is, we're really enthusiastic right now about some of the positron emission tomography work that we're doing in our lab, but that's never going to be a really useful biomarker for many contexts in autism because it involves administration of radiation, right? So we wouldn't do it in young kids. There are biomarkers that are useful, biomarker acquisition modalities that could be more useful. We would want biomarker technologies that are cost effective. The expense of administration is going to be a ceiling on deployability and scalability for any biomarker. And likewise, we would want ones that are accessible. Like if we had, for example, a biomarker that was useful but required such specialized equipment that it could only be done at an ACE center, right? This would be its reach. And I'm in this crowd, I probably don't need to define it, but ACE Autism Center of Excellence, our major NIH-funded autism research institution. This would be its reach. If we could, deploy a biomarker at every hospital, this would be its reach. And clearly, this is what we would want if we were to impact actual clinical care in autism. This is a biomarker acquisition modality that I'll talk about today, one that I do a lot of work with. I saw Sophie in the audience. Sophie is another exquisite EEG biomarker researcher. EEG stands for it means electroencephalogram. This is the electrical activity produced by neurons recorded directly from the scalp. Whenever we do anything, our neurons produce a small amount of electricity. If enough of them do that in unison with a particular orientation to the scalp, we could actually measure that from the scalp. In general, we do this in two situations. One, when a person is just sitting there and we can measure their kind of oscillatory idle of the brain. Two, when we make things happen, like we play sounds repetitively or we show things in the environment repetitively, and then we can measure the response that is elicited by those perceptual events, and we can determine then what the brain is doing in response to one of these specific things. So we can learn about how the brain is responding to the sound of a voice or to the sight of a face, for example. This is the technology we use. There's different technologies. It's technology I use in my group and in the consortium that I'll describe. It's called the Geodesic Sensor Net. This is an elastic mesh. Each little circle you see is a small rubber pedestal with a sponge embedded in it and an electrical sensor at the base of that sponge. You dip the entire contraption into salt water. So the sponges absorb the salt water. They become conductive. You stretch it over a person's scalp. The sponge is making an electrical bridge between the scalp and the sensor, and so we can pick up that electrical signal. Because this technology just requires wearing this damp, wet hat, it's pretty user-friendly in terms of brain measurement. We can do it in intellectually gifted people. We can do it in intellectually impaired people. We can do it in the elderly. We can do it in infants. It's non-invasive, and compared to other forms of measuring brain activity, it's relatively movement tolerant. So it's a viable technology to use in autism. It's practical technology. It's expensive to buy an EEG system, but when you have one, collecting data is the cost of staff time and salt water. It's an accessible technology. We have EEG systems in most hospitals in this country. This is a technology that's already used for screening at the population level for seizures and for newborn hearing. And so there's an infrastructure in existence that if we should learn something useful, it could be deployed cost effectively in its scale with relative ease. And it's also for for studying social communication, which is one of the few things that we know is a universal that we can presume to be true of a person diagnosed with autism, it's actually very, very effectively applied in non-autistic development so that we have a frame of reference and a foundation of understanding to try to understand how things might be developing differently in autism. Okay, that's kind of the background about biomarkers and this method. And now I want to talk about kind of a particular biomarker, the N170. And so this story starts when I was in graduate school. My advisor, Jerry Dawson, said, hey, why don't you look into faces? And this made a lot of sense in autism. Faces are the first, maybe one of the first sources of social information that we get. We know a lot about the neural systems that support them. They can be measured in a very specific way with EEG. There's this index called an N170 that is telling us that the brain is registering a face. We think of faces and face-related activity as potentially meaningful in autism because people with autism are so heterogeneous. In this group of expert diagnosticians, you will know that there's only one thing that you can know is true behaviorally for a person with autism, and that's they have differences in social communication, right? That's a non-negotiable when we're making a diagnosis. And within that, there's lots that's negotiable, but we know that face and gaze processing differences are something really common in autism. So maybe this is a way that we could go with biomarker discovery. And so the first study I did when I was in grad school was to look at the neural response to faces in autism. And this is what seeing here. This blue line is an event-related potential. Event-related potential is just a way of processing the EEG signal to tell you the information that is specific to a particular percept, in this case, a face. So this is the N170 that I mentioned. You can see this negative spike highlighted in purple is the N170. It's called an N170 because it's negative going electrically, and it's happening at around 170 milliseconds. When you look at this chart, your y-axis is the strength of your signal measured in microvolts. Your x-axis is your time in milliseconds. Note that this is really fast. This entire chart is just 7/10 of a second. And so when I tell you this is an N170, that's your brain recognizing a face as a face in less than 2/10 of a second. We did this first study in autism, and this was following on Bob Schultz's work with imaging, and we wondered if we'd see the same thing, that maybe there'd be an attenuation of response, and there wasn't in the same way. The difference that we saw with the temporal sensitivity of EEG was a difference in timing, that we saw that this brain response was slower in autism, about 10% slower at this very early stage. So this was the first in a series of studies that we've done in our research group to try to understand whether the N170 is reliably different in autism and how it might map onto other biomarker properties, the ones that I mentioned before that are relevant. And as I go along and talk about these studies, I'm going to refer back to that list. And so going back to that list, this is telling us here about something about diagnostic status, that not to the individual, but in general, we're seeing that this N170 difference is slower in autism. It hangs together with diagnostic status. In this same study, we wanted to get a sense of whether it might also map to any functional domains, and so we looked at face recognition. Behaviorally, we found what many studies have found is that the autistic sample had more trouble with face recognition. They made many more errors compared to a cognitively matched sample. And we also saw that the number of errors they made was associated with the latency of their N170. So we thought, okay, well, we see this thing that's different in autism, and then we also see that it seems to hang together with something that we think could be clinically relevant, face recognition. We having discovered this basic difference, we then have done a series of studies to kind of understand more about it. One thing that I wanted to understand, especially at the time that we were doing this work, there was a lot of kind of neuroscientific debate about whether the differences that we see in autism are reflective of kind of differences in social systems, or whether they might reflect differences in processing complexity, or what if it's just a brain that processes all sensory information more slowly. And so what we want to see is that this slowness that we see for faces isn't a universal, that it's really specific to faces. But that's a tricky thing to measure because then you have to find something that elicits similar brain activity and show that it's normative. And so we got this idea from this kid that I worked with at the time. When this video was taking, Celine was probably behind the mirror supervising me on this day. But this is, you'll see from an
These
ANS video.
W's looks like they're on top of each other.
They sure do. You a picture?
A picture?
Yeah.
WPS.
Oh, that's what you remember that.
Hey, that boy's not underwater.
He's got a mask and a snorkel.
And it looks just like a J.
You're right, it does look like a J.
So you all are expert clinicians. You're probably recognizing that this is a young boy with autism who has hyperlexia. He's super duper into letters. It actually turns out that when you learn to read the alphabet, any alphabet, your neural systems kind of recruit this face processing system in a different way, in a left lateralized way. But you actually see an N170 to letters as you learn to read an alphabet. So this gave us this idea, well, we could use this to see whether the differences we've observed to faces in autism are specific. And if this is really purely a social difference, when we look at brain responses to faces, we would see the slowing we've seen, but we wouldn't see it when we compared letters to a made-up alphabet of pseudo letters. In contrast, if this is just generic slowing or a generic problem with specialization, we would expect to see differences in both areas. So we also use this as an opportunity to understand something about the development of the N170 and looked at kids who were a little younger. So these were school-age kids compared to the adolescents and adults that we'd seen the initial differences in. And when we looked at the faces, we saw the same kind of things that we saw last time. We saw that the autistic participants made more errors with face recognition, and we saw that their N170 was slower. So this is good. We're seeing that it's replicating in a younger cohort, and we're seeing this one biomarker seems to show the same kinds of effects relative to non-autistic people across, from like age 6 or so to age 42. When we looked at the non-social information, the letters and the pseudo letters, behaviorally, we measured behavior here by looking both at word reading and phoneme decoding, and we saw that there weren't group differences. The autistic children were average and were comparable to the neurotypical peers. And when we looked at their brain responses, we saw two things. We saw that both groups showed specialization. So this is the amplitude, the strength of signal of the N170, and the purple bars are bigger than the green bars. So it's showing us brain response that's reflecting specialization for letters. And so, so we see the brain specializing as they should. In terms of latency, we didn't see differences in latency. We saw that they specialized as we would expect. Specialized in a way similar to the neurotypical people. Only difference we saw between the groups was that the autistic group seemed to involve the right hemisphere that typically involved in face processing more when they were viewing letters relative to the non-autistic participants. So this suggested to us that, okay, well, this thing that we've measured, that we've seen is different than autism, that is related to behaviors that are clinically relevant, that is consistent from childhood through adulthood, also seems to be telling us something specific to the social domain, which we were thinking is relevant to autism. The next question that we asked was whether the way that we collect the information could be influencing our results. So it turns out that the N170 is influenced by the part of the face that you look at. And specifically, when you direct a person's gaze to the eyes, you see the N170 get faster. So we know both clinically, anecdotally, and from a long series of eye tracking studies done over the past 20 years, that people on the spectrum look less at the eyes. So it's possible this N170 difference that we're seeing is really just telling us that when you put an autistic person in front of a computer screen, measure brain activity, you show faces, maybe they're not looking to the eyes. Maybe they're looking at other parts of the face, and maybe that's why their N170 is slower. And that's fine, but then we don't need to bother with EEG, right? We could just be then the biomarker is eye track, and we don't need EEG. And so to understand this, this was work led by Tamara Parker, who was a graduate student at the time. Um, we did an experiment where we had a crosshair that would manipulate visual attention. So what you're seeing is not veridical. The crosshair didn't co-occur with the face; it preceded the face, but there'd be a crosshair on screen, then the face would appear such that a person would be looking to the eyes, looking to the nose, or looking to the mouth. And what we wanted to understand was, if we make everybody look to the eyes, do these differences that we've seen in autism go away? Is it just that they're not looking to the eyes? And that wasn't the case. So what you see here on the y-axis is your N170 latency. You see three groupings on the x-axis when people are looking at the eyes, when people are looking at the nose, and when people are looking at the mouth. The yellow dots are autistic participants. The blue dots are non-autistic participants. And those gray diamonds are showing you the group average. And what you can see, so I don't depict it here, but if we were just smearing this all together as if we didn't have a crosshair, you'd see overall a slower latency in autism. But as we look in this more granular way, we see that not only does looking to the eyes not eliminate the differences we see in autism, that's the place where you see the differences, is that people with autism show a slower N170 when they're looking to the eyes. So this is not just an artifact of behavior when they're coming in to do an EEG experiment. It's telling us something that is true, reflects a true difference in the brain when we have reasonable assurance that the same information is getting to the retina. Okay, the last study that we did in this kind of exploration, and this is one that I should couch as preliminary because it's such a small sample, but it's also a rare sample in that there are astonishingly few studies that have looked at the N170 before and after change associated with behavioral treatment. And so what we did here, this is an opportunity study. Pam Ventola at the Child Study Center at the time was running a PRT study, 14-week course of treatment. As you know, pivotal PRT is pivotal response treatment, and empirically supported naturalistic behavior intervention for autism. In this study, she treated the kids for 14 weeks. They got better. We measured their N170 before this treatment and after this treatment so that we could get a sense of whether the N170 might change. And what we specifically wondered was, as these kids get better, do their N170s get faster? And so they did. These are data from the individual children. There were only 7. Each line represents a child. The dotted line represents the average. At left is before treatment, at right is after treatment, and on the y-axis again is N170 latency. And so when you see the majority of these lines sloping downward, that is telling us that their N170 latency was shorter after treatment than before treatment. Not true for everyone. You can see participant number 3 actually didn't change or got a little bit slower, actually. But in general, we're seeing in this small preliminary sample, N170 latency is, as a participant's behavior moves more towards what we would expect in non-autistic development, we're seeing their N170 also more move towards the direction we would expect in non-autistic development. So we did this series of studies, right? These studies range from a sample size of maybe 23 to 60 or 70, and we saw these things. We saw hits across these different my different report card categories. It hangs together with diagnostic status. It associates with clinically relevant functions in a way that we can presume is relatively specific. We see it consistent across development. We see it's robust to variation in behavior during biomarker acquisition. Preliminary data suggest it's sensitive to clinical data, to changes in clinical status. And then we see, because we're using EEG, it's with a technology that's viable, is cheap, and is accessible. So we have a decent body of evidence suggesting there could be some there, but how do we get to a place where we could say, all right, maybe this is something that we could actually use in clinical research or in clinical practice. And so one way to do that would be to do a different kind of study, a more rigorous study, to demonstrate that we can replicate these findings at scale across sites, to understand something about reliability. In none of the studies that I have described did we measure the same thing in a person more than once outside of the context of treatment, which is critical for understanding whether there's something stable of a person. Across the studies that I've described, and I didn't summarize, but there are many studies. By now, I'd say, I don't know, 50 or 60 studies of the N170 in labs other than my own. The majority have replicated. Meta-analyses have suggested this is consistent across the studies, but it's not universally true, and we don't know why. Could be differences in samples, could be differences in methodology, like whether you use neutral faces, emotional faces, crosshair, no crosshair. So we could do a study where we really control for those kinds of things. So we eliminate methodological noise. We can have really deep clinical phenotyping so we can understand more about potential relationships to the phenotype other than what I've told you so far, which is just face recognition. And we could have large samples so that we have the statistical power to make strong inferences in these kinds of domains. And these are the considerations that were bandied about in the kind of early 2010s in a series of think tanks at NIH that I wasn't involved with, but then begat the RFA to call for a consortium for biomarker development in autism. And that RFA then gave us the opportunity to start the Autism Biomarkers Consortium for Clinical Trials, which was a study really designed to enhance rigor across these different areas. The design of the ABCCT was it was a multi-site naturalistic biomarker development study. So by naturalistic, I mean that it wasn't a clinical trial. There was no intervention introduced. We just measured intervention that children were receiving. We took well-evidenced biomarkers, and so we would have clear hypotheses about what we should see, and then sought reproducibility. We had large samples, including large samples of non-autistic children, deeply phenotyped cohorts, a longitudinal design, a baseline, a 6-week visit to look at stability, a 6-month visit to measure kind of change or stability in the context of a standardized clinical trial, and then a more distal longitudinal follow-up 2.5 to 4 years out. We use practical assays, EEG, which I've talked about, but also eye tracking, which is also a pretty cheap and deployable technology. We held ourselves to high methodological and regulatory standards, administered the study as if it were a clinical trial according to good clinical practice. And then we collected genetic information so that we have that information for the future. There were 2 phases to the ABCCT. Data collection is now done. The first phase was 399 children in between 2015 to 2020, and then over the past 5 years, we saw another 450 children. So these the assays were consistent, so these can all be combined into one large sample. I won't go through them, and I'm happy to share my slides afterwards, but you can see here we did like the status quo in terms of clinical measures used diagnostically and in clinical trials in autism. And then we had this battery of biomarker assays that I won't go through, but we did include this N170 assay that I've been talking about so far. When you see an asterisk here, that means this is an assay that we worked with our colleagues in Europe initially in EU AIMS, and when they were renewed, they changed the name to AIMS to trials so that we have replication samples across these 2 consortium for the biomarkers that are asterisked. There you see Sarah Webb depicted, who has been the director of our data analysis and acquisition core in the ABCCT, and really has done an incredibly thorough and remarkable and rigorous job of understanding these data. What we learned with Sarah in terms of this N170 was that we saw replication. In this sample, we saw the same kinds of effects that we've seen before, this slower latency in this larger sample of people with autism. We saw that it was a feasible assay to deploy across multiple sites. All these sites were experienced in EEG, but We got data from 97% of the non-autistic kids and three-quarters of the autistic children. We didn't see differences across sites. Um, ac- we expect certain changes with age, but we didn't see confounds with age or IQ either. And we also saw, really importantly, that our colleagues in Europe saw the same pattern of results in their data. We learned something new, that these indices are relatively reliable within a person, both at 6 weeks and at 24 weeks. We saw that they were related to the phenotype, but in a very specific way. Even having all these different clinical measures, we saw that this output of the face processing system really specifically associated with face recognition. So in a very specific way, we learned that it's very important to control for the way we control for age when we're doing a Vineland or when we're doing an IQ test. We want to control for age too when we're measuring output of the face processing system. And we one of the problems, that not a problem, but like a bad luck of this study is that measuring treatment as usual in school-age children, we really didn't see a lot of kids get a lot better or a lot worse. And so we really don't have variance in this study to understand a lot about how these biomarkers might relate to changes in clinical status. With that information, we then tried to kind of engage with the FDA. The way it works with the FDA biomarker qualification program is the first step of 3 is to submit a letter of intent, basically showing them your data and saying how you think it could be used. What we said is that we think this would be basically a stratification biomarker, diagnostic of a subgroup, that like when we look at these distributions, autism in green, non-autism in blue, you can see this tail really only exists in autism. And so we said, well, maybe this is a subgroup that we could use to enrich clinical trials by picking a more biologically homogeneous subgroup. The FDA bought it. They accepted our letter of intent. This was in 2019. I haven't gone into it. We also had a letter of intent accepted for an eye tracking index in 2020. This is the first of 3 stages. It's the easiest of 3 stages, but I think it's also an important milestone because it's the first time that a biomarker for a psychiatric or neurodevelopmental condition like autism has been considered by the FDA. And that's really where things stand in terms of the process of regulatory development. We continue to work in coordination with our colleagues in Europe who are doing the same process with the European Medicines Agency and are at the same stage. And then other collaborators as well, Evdokia and Agnostow, and the PON network collaborators in industry, really to see how we can create a roadmap for something that really hasn't been done by academics nor by the FDA, to figure out what kind of data could be adequate to advance a psychiatric biomarker to the next stages. Okay, I want to talk now about work in progress that kind of is seeking to make this biomarker more relevant. Not this biomarker, but this and other biomarkers more relevant and potentially more applicable to a wider portion of the autism spectrum. So one thing that we're doing is trying to understand how it actually works in more realistic life. When we do an EEG, it's a person interacting with a computer screen, which is not social. We can use other methods like functional near infrared spectroscopy to actually record brain activity while people are interacting with one another. And this doesn't tell us about the N170, but it tells us about metabolic activity, oxygen consumption in the same kinds of brain regions that we think generate the N170. And what we've seen here is that we see very specific responses associated with in vivo eye contact that really correlate with the ADAS. So this idea, even aside from an N170 specific assay, looking at the face processing system seems to have legs in more realistic contexts. We're thinking about it also as a potential avenue for intervention. I mean that in the loosest and broadest way, but we can actually, if we have a neural system that we're measuring that seems to be inefficient in some way, we have technologies that could induce plasticity or stimulate that system. And so this paddle here it's using a technique called transcranial magnetic stimulation, which is exactly what it sounds like: powerful magnetic pulses that induce action potentials in neurons close to the skull. And what we can then do is target a certain region, like the superior temporal sulcus, which is relevant to face and biological motion perception, and then stimulate it and see the relationship between the biomarkers and this stimulation. And what we've found so far, and this is work I'll highlight, done by Sherab Shloringa, Azia Alsop, and Jacob Momsen, and others in the lab. And what we see is that we're in the early stages of understanding these data, but what we see so far is that when you stimulate this part of the brain, we see with eye tracking, we see more attention to the eyes in both autistic and non-autistic people. And what we see for the N170 is, well, one, we see replication again. When we're not stimulating, we see the autistic people have a slower N170. But then when we stimulate, we're seeing a very specific effect, and that is an increasing of N170 latency when they're looking to the eyes in the autistic participants. So what does this mean? We don't know yet. Could this be telling us who might benefit? Like, are there people who would experience increased comfort, for example, making eye contact with repeated stimulations? Might this tell us about that subgroup of people who would benefit from a treatment? Might it tell us that a treatment is working before we would see behavioral differences? Those are things that we have to gather the data to understand. We're also trying to do a better job of being inclusive on the spectrum. So in this ABCCT, I didn't mention it, but every participant in the ABCCT had an IQ above 60. We know that that's missing a really important segment of the spectrum. So Adam Naples in the lab and Christine Sugar-Capizzi, who's a BCBA, have worked together to develop both kind of hardware and behavioral solutions to help support much more cognitively impaired people with autism engage in neuroscience research. What we've done is a few things. One, Christine has really works with each family to develop a child-specific behavioral protocol for desensitization to the lab and to kind of set up reinforcements to get them through the experiments. And what Adam has done is build kind of an automated system that measures what a person is doing using motion sensitive cameras, and basically sets up a set of contingencies to shape sitting still and attending to the screen, and then also to just follow a child's lead and only administer the experiment when they're paying attention. And with these kinds of approaches, we've experienced great success. We're running studies now in children as young as 3 and as old as 17, where we're seeing that we're getting valid data in people with an average IQ of 31 in some of these age ranges. We see construct validity in terms of the N170 being face responsive. And then we also see, so far, we see in this preliminary sample that the N170 delays that we've seen seem to be more pronounced in the sample with both autism and intellectual disability. We're trying to create, and this is to do more work with different kinds of markers. One approach is basically to take a page out of mouse researchers who use cameras in cages to measure mouse movement, and that's a way of generating behavioral biomarkers. And so we can have a play session here where where we can measure a person's movement in the room. These are not data from our lab, but from Ira Cohen, who's been a collaborator in the ABCCT. And use this basically as a measure, as a proxy for social engagement, showing that, for example, if we have a parent in the corner of the room and toys around the room, neurotypical children are more likely to kind of stay in the geographic vicinity of the parent, but the person with autism is more likely to stay near the toys, or less likely to come near the parents. We're trying to do more with these data. Jacob Momsen is doing, trying to apply AIs to ADAS videos in a few different ways. One is just to measure in a theory agnostic way overall movement and acoustic properties of the participant during the ADAS. And what we find is that what we expect is that people tend to move and use speech in coordination. And we see this less in proportion to more autism features. This is something that we're really just kind of beginning to play around with. But I think the theme of this and the next two slides are really that I think that we now have tools, AI-based automated tools to use the information that we generate during the ADAS and other behavioral assays in ways that really haven't been effectively leveraged historically. This is one. Another way is to create transcripts of language during the ADAS and use large language models to look at word choice. This is work of Bree Kearney in the lab, who finds that when we look at kind of, historically we called it idiosyncratic language. She would call it rare or unique word usage, but that we see much higher levels during the ADAS storybook task in the autistic group, a greater diversity of language. And then, Bree is doing this in a theory-driven way to kind of look specifically at these word use, but Billy Carson is a computer scientist who's doing it in a much more data-driven way, really, again, using AI to basically look for relationships between the language that's happening in the ADAS and attributes of the people. This is just an example from the first pass of the data that he's run. And if you look at these kind of dots, each of these dots is a person, and the color is their age. And what you can see is that these darker dots tend to hang together. And what this is telling us is that people who are older are using language in ways that are more similar than people who are younger, which is intuitively appealing and not clearly clinically useful, but it's also something that we wouldn't have hypothesized. And so when we apply these tools to these rich data sets, we will have the opportunity to generate new hypotheses and to learn. Okay, that's what I want to talk about. We have a few minutes for questions. I just want to thank, most importantly, the kids, adults, and families who come in and do this work with us, the people in the clinic who really that is where our relationship with the community is founded and where the work starts, the investigators throughout the ABCCT, and then the people in the lab who do lots of this work. Okay, and I will stop my share so I can see if there are questions, and I will lower my own hand so you can raise yours. Jim.
This was fantastic, Kenny. Really great to see all this work. I was curious if you can speak to like the difference between like showing a group difference versus building a biomarker that would be specific for diagnostic purposes, let's say. Maybe just speak to what that would entail at that kind of progression from group difference to an actual, clinically useful marker.
Yeah, I mean, some of these questions are like, that's a great question for like an epidemiologist, right? Like you think like when you say that, I think like a positive predictive value, right? And so I didn't make this comment when I had up that slide, right? But like, so like for those of you who don't spend a lot of time thinking about this, like think about that slide. Maybe I'll just show it again. But like when I show this, so if you look at these distributions, like this is useless as a diagnostic tool, right? Because most is overlap. So like now, so Jim, if we had like the opposite, if the autism distribution started here, right, that'd be pretty awesome. I have never seen anything like that in neuroscience, except for once when I was writing a grant, and in the preliminary data sample size of 7, we had non-overlapping distributions, which I think is reflective more of an idiosyncratic sample than truth. So it's part of the reason, like when I talk about the work that I do, is I think that stratification biomarkers are more reasonable, attainable goals. It's hard for me to imagine, like when we talk about autism, we are not talking about a biological thing, right? We're talking about a diverse constellation of behaviors seen over a wide span of developmental time that we happen to see in one person that lets us put a person into this category. But the idea that we can capture something biological that is true to the person rather than that category is a really tall order. So I don't know if I don't think I really answered your question. Um, I don't know. I don't know how to do it. I don't know that it will be done. I think that the way that Ami and Warren and others are thinking about it as like an add-on or like a supplement to the diagnostic process is the best way to think about it. But I don't think we're close to having a place where there is something that is biologically true of idiopathic autism. Maybe the exception will be, like more biologically defined subtypes like neurogenetic syndromes or things like that. But that's like the $1 million question, I think.
I realize we're up against time right now, and we have one question in chat. But I appreciate your last comment because I think that's what everyone's kind of leaning towards now is what is not a distinct biomarker that separates, diagnostic categories, but instead identifies biological processes that can help us make decisions about overall neurodevelopmental disorder that some of us call autism. Jan, I'm just trying to process everything you just threw at us. It's like a fire hose of stuff. So I'm having a hard time, like even coming up with a question or a comment, except that to end with this is like what I really appreciate about your work is it's not only informing basic science and understanding autism, but neurodevelopment in general, but also you're making moves towards application and what this might look like in the real world, which I think very few labs are doing and doing well with the kind of rigor that you do. So please don't stop the work that you're doing. It's incredible. Keep publishing, keep working. Everyone join us next month when we have Celine and Cynthia teaching us how to make money instead of doing what we're doing now. Jamie, do not go to that talk because we stay in the lab figuring this all out for us. But thank you everybody for joining. Stay warm, stay safe, and we'll see you next month in February. Bye, everybody. Thanks, Jamie.
Thanks for having me, Steve. And thanks for clarifying. I hit my goal, which was volume of information at the expense of clarity or comprehensibility. I nailed it.
Nailed it. Crushed it.
All right. I'll see everybody later. And
It
Theo,
was actually very
Theo,
clear.
there is if Theo is still on, there is like a long and complicated answer to your question, but
I will let you guys deal with that yourselves.
he or she or they is not on, but I'm gonna email them.
Okay.
All right. Awesome.
Take care, guys. Bye.
All right. Thanks, everybody. Bye-bye.